Okay. Hi, everybody. We're just going to give it about a minute for everyone to kind of finish joining. So we'll start in about a minute or so. Okay, I think we're probably gonna get going. It seems like everyone's joined us. So I'd like to welcome everyone to our second webinar so far. Today we're going to be discussing COVID testing, expanding your lab capabilities. And I hope everyone can see my screen very well. And if not, please let us know in the chat if there's any issues. So first off, I'll start with introducing myself. My name is Carolina Corlin, and I am the marketing director here at PBI. We're going to cover a little bit of housekeeping before we begin. So the webinar, it is being recorded, and the recording will be available on our website, and also a link will be emailed to you afterwards. That will probably be closer to the end of the day. Everyone is on mute, so just so that you know, there won't be any background noise or anything like that. And the webinar itself will be about 40 minutes. There will be a Q&A session at the end. And so we do encourage you to ask questions, but please use the question tab. And so it's the little kind of exclamation mark or question mark in the bubble that says question. So please write your questions and note your questions there, and then we'll address them during the Q&A. So that's it for our housekeeping. And in terms of our learning objectives for today, we will be reviewing the PCR testing. So we'll review the details of real-time RT-qPCR testing and highlight how it is suited for laboratory settings. The second objective will be to cover key considerations for selecting an assay. So that's highlighting key features and aspects of a test to consider when selecting a COVID-19 assay and how it can affect your testing strategy. And then number 3 will be the ways to streamline the lab processes. And that will be a review of the features that will aid in making your laboratory run more efficiently while meeting COVID-19 testing demands. And so our presenter today is the lovely Amanda Naaum, who is who has been with PBI, who has over 10 years of experience in molecular assay development for species identified in both academic and startup industry settings. She does hold a PhD and has been awarded with the IUFOST Young Scientist Award and the Commonwealth Rutherford Fellowship in recognition of her research and innovation achievements in point-of-need food species authenticity testing. She now extends this expertise to applications for food and animal safety, environmental monitoring, and human diagnostics here at PBI. And she's also the co-founder as well as the Scientific Liaison Director. And a little bit about us in terms of who we are. So Precision Biomonitoring was founded in 2016 by a team of scientists from the University of Guelph's Biodiversity Institute of Ontario. Our very own triple lock technology delivers on-site eDNA surveillance and human diagnostic test solutions. We provide detection tools for various scenarios ranging from laboratories to rapid, accurate on-site organisms and virus detection tests, including COVID-19. To support the pandemic response, we offer industry-leading testing solutions approved by Health Canada, and the following tests are lab solutions, which are shelf-stable PCR COVID-19 testing plates for lab settings, which is what we'll be discussing in greater depth today. And of course, our mobile solution, which is also a rapid, accurate point of need on-site qPCR test. And so without further delay, we'll jump right into it, and we'll start with our first slide on testing and expanded COVID-19 testing, expand your lab capabilities by reviewing PCR testing itself. Pass it over to Amanda now.
Thanks, Carolina. Do you know if it's possible to hide our videos? Because I'm still seeing them over top of the slides, which...
I think... sorry, 1 second. I don't know if it's just you and I seeing this, or if this is everyone else, to be honest. I think it might be...
I think it's everyone.
Is it everyone? I'm sorry. Okay, yes. Maybe if we turn off our videos, but I'm not sure how.
Yeah, I turned off mine, but I think I still see just the empty slot for my slot.
Yeah, I see this empty slot too. I apologize about this. Yeah, it still blocks the slide. It looks like. I'm gonna see if I can. How about this? Does this help? Oh no, but I think now I've... Amanda. Sorry about this. We are having some technical issues. Amanda?
Yep.
Okay. Sorry, I'm not sure how to actually do that. And I know that it's maybe if we can, Amanda, why don't you start, and I can maybe fiddle around with some of the settings here and see if we can make that go away.
Okay. I think that the majority of the content is text, which is great because you'll get a copy of the actual slides, I think, later, and hopefully the actual recording will give you that information. So you can hopefully bear with me, and I'll go through all of the content here as we go. What is PCR testing is kind of our first segue into the learning objectives that Carolina has just gone over. So PCR stands for polymerase chain reaction, and it's essentially a molecular test method that's used to analyze or detect really a short sequence of DNA. And the way that it works in a nutshell is that you design a test to look for a specific target, and that target is amplified using a bunch of heating and cooling steps to make lots of copies, and then the test detects that DNA. So for real-time PCR, which is commonly used for COVID testing and other forms of diagnostic testing, really the analysis of the presence of that short target sequence that's being amplified is obtained in real time, and it's based on fluorescence monitoring. So there's all kinds of different ways that you can assess the presence or absence of that target depending on the kind of PCR. If you have a look at the image to the right, that kind of goes through the steps for a real-time PCR reaction. So PCR essentially makes use of enzymatic function to copy a specific target, and that target is identified using what's called primers, which we're going to be using that term throughout the presentation here. So the two primers kind of flank the region of interest, and they're specific to that specific target that you're looking for. Then in the case of real-time PCR, you'll see in the middle there is a fluorescent probe that also attaches in between those two primers. And as DNA polymerase extends the single strands of DNA during the PCR reaction, that probe's broken apart, and that fluorescence is actually released and can be read. So that's kind of the real-time portion of real-time PCR, so that fluorescence is then monitored over the course of the reaction. So to detect RNA, we would use reverse transcription PCR, which is exactly the same as what I've just described, except that first the RNA within a sample is converted into DNA using reverse transcription and another enzymatic reaction, essentially. So that step happens first, and then the presence for a specific target is administered. So these tests are usually highly sensitive and can detect even low levels of genetic material. And they can be used for all kinds of different organism identification, including bacteria and viruses. They also are used in terms of kind of PCR generally, used for all kinds of other applications where you want to copy a lot of a certain target before you run analyses. And that could be DNA fingerprinting or sequencing, as well as diagnosis of genetic disorders. So kind of copying what you have first so that it's more easily detected. So on the next slide, so PCR testing for COVID-19 specifically is designed to look for the COVID-19, so the SARS-CoV-2 viruses, genetic material. So those primers are specific then to that. Since coronavirus don't actually have DNA, their genetic material is RNA. We use that reverse transcription step as a step before PCR to create DNA to then be able to be detected. So the PCR machine, by means of heating and cooling over many cycles, like a washing machine might do, amplifies up that target if it's present there. So as you have more cycles run, there's more copies of that DNA made. So if you think about in the previous slide with the image of splitting, all of the genetic material apart and then making a second copy of it, basically what you're doing is doubling the amount of that target that's present every single cycle. So that just makes it easier to detect and easier to find if it's there in really small quantities to begin with. You can imagine kind of after that exponential amplification over 35 or 40 cycles, you got a lot more than what you started with. So if that piece of DNA cannot be copied or cannot then be detected at the end of PCR, then either there's no virus in the sample or there's a very low amount such that the test actually can't detect it. So that's kind of the principles behind how this is applied to COVID testing. So for positive PCR tests and kind of how to interpret them, a PCR, or in this case, I'm going to use it interchangeably with real-time PCR just because that's what is typically used for COVID-19. So real-time PCR will tell you if the virus has been detected, so whether it's there or not. So you've got either a positive or a negative for the most part for COVID tests. Real-time PCR can be quantitative, but typically that's not the type of testing that's employed for COVID-19. Often these tests will have a cutoff point for positive results. So you may run the test for 40 cycles, but you may have a cutoff for positives that is 35 cycles, 38 cycles, maybe it's 40 if you're running for 45. And that cutoff point really gives you an idea of where the confidence of a positive result kind of cuts off at, just because there can be non-specific amplification sometimes beyond that point. So with that in mind, it's important to note that different PCR tests would have different cutoff values, and that might be based on how sensitive the test is, how it's been designed, the type of validation that was done. So it's really important to follow the instructions for use for those particular cutoffs, and provide feedback on that based on your testing as well. And there's laboratories across the province and country that use different testing kits. So there are different tests available to test for COVID-19. So PCR testing, or polymerase chain reaction testing, was actually developed over 35 years ago. As I kind of mentioned a little bit in the first slide, it's one of the most widely used lab tests for a lot of different kinds of things, and in particular for finding viruses and other pathogens that cause diseases. This really is one of the gold standard tests for doing this type of detection and diagnosis. So the World Health Organization has recommended nucleic acid amplification testing, including PCR as one of those, as a preferred testing method for COVID-19. And that testing will essentially tell us if someone's infected with the virus, as mentioned. So this quote from Public Health Ontario just notes that in Ontario, PCR is the gold standard for testing for COVID-19 because it's able to successfully identify small amounts of the virus with a low chance of error compared to other lab tests. So in Canada, at the federal level, PHAC leads Canada's public health response to COVID-19, and they work with public health laboratories across the nation to test specimens received from provinces and territories, as well as to develop accurate tests. And PHAC also publishes the number of people tested in Canada every day through that system. At the provincial and territorial level, provinces and territories are responsible for the delivery and administration of their healthcare services, and that includes the public and private laboratory developed tests as well. So they can develop their own sample collection, testing methods, responsible then also for ensuring that those test results provide reliable results. So that includes ensuring that laboratory developed tests and collection methods are safe and effective. And these laboratories that develop their own tests or offering testing services are not necessarily regulated by Health Canada. So the development of those tests doesn't fall under the same regulations as some commercially available tests might. So that's kind of an overview of PCR and kind of PCR for COVID specifically. The next few slides are just talking about how you might be able to expand lab capabilities and considerations for selecting an assay to do that. So on the performance end, kind of had mentioned this a little bit within requirements for some of the lab developed testing, but you want to have a test that's both highly sensitive and highly specific. So sensitivity refers to the ability of a test to identify people with the disease as actually correctly having the disease. So that is a measure of true positives. Specificity, by contrast, is the opposite, which is ability of the test to correctly identify people without the disease, so your true negatives. Related to specificity, all tests should include some assessment of cross-reactivity with both closely related organisms or co-occurring pathogens. So for example, in the case of a COVID-19 test, you would want to have some cross-reactivity data on not only other coronaviruses, because they're closely related, but also other respiratory pathogens or organisms that are likely to be present in the sample type that you're collecting. So if it's a nasal sample, that would be other respiratory organisms. For tests that are working on a saliva-based collection, if that's something that your PCR test is designed to work with, that list of cross-reactivity organisms would be different because you're wanting to test against other things you might find in your mouth instead of your nose. And luckily, the list is not overlapping entirely. So that performance of the test can then be affected by the sensitivity of the test itself and specificity, but also some items that actually have nothing to do with the performance of the test in terms of the actual PCR portion. And that part is related to how the sample is collected. So are you using the correct sample type? Is it being collected properly? the actual preparation of the sample. So has the extraction proceeded accurately? Were there any challenges with yield or inhibition? And then potential for contamination as well would have to be considered within that sample preparation. So this also includes the timing of the sample, in the sense that you want to collect at a time where the amount of viral load is actually going to be detected, which can change across various points of the infection. So this can give you really, this is to say that it's going to give you a snapshot in time. A PCR test will tell you the viral load of that sample at the time it was collected, and also is contingent on a properly collected and extracted sample as well. So it's only going to be as good as the sample that actually went into the test. So false negative results, where you have somebody labeled not infected when actually in fact that they are, generally are considered more consequential than a false positive, where someone's told they are positive when they're not. And quite a lot of this possibility for both false negative and positives can be attributed to the factors above. So it's really important in test collection to have a good understanding of how do you prepare a sample to work appropriately with that PCR test, and that you have trained personnel for sample collection and processing, as well as running the test itself. So in terms of interpreting results, this is starting to get into the technical side of the test and PCR in general. But for real-time PCR, I mentioned earlier, this is based on fluorescence output over a number of cycles of heating and cooling. So a cycle threshold, or CT value, is the actual number of cycles that it takes for that fluorescence to exceed a given threshold. And that kind of indicates that the target is present. So if you have a think about the graph that or diagram really that was presented earlier about that copying over time and doubling over time, you can imagine that you're talking about fluorescence output over these numbers of cycles, that the earlier that you have this crossing threshold, so the earlier that you kind of hit that minimum amount of fluorescence, means that you had more starting material of that target present. So what that means is, in very general terms, again, because this is mostly not quantitative assays per se, but they're semi-quantitative in the sense that if you start out with a lot more of the target, you're likely to be getting that fluorescence coming up earlier in your number of cycles. And so this image just kind of gives you an idea of what that typical output might look like and where that CT value would be calculated from. So you've got fluorescence on the y-axis and number of cycles, or essentially time across the x. The blue line represents what a positive sample would look like, with the green kind of vertical line coming down at that crossing threshold. So the black line is sort of that threshold setting. That threshold setting will either be automatically calculated by the machine or manually set based on the assessment of the test. So that's something else that can vary depending on the test. So you want to make sure whatever test that you're selecting, you have an understanding of how that threshold should be set, because it can adjust results for sure. And then the red line kind of shows if there was no template. So if there was no target actually present, you get this negative curve, sort of a flat curve, where you're not seeing any fluorescence buildup. So that's kind of the ideal scenario, and how positive samples are differentiated from negative ones. And that CT value kind of comes into play with that, where you might have a sample that has some really late amplification, and so you can get those CT values over the cutoff value that we had mentioned earlier that some tests might have. So that's, again, to just account for potential for non-specific amplification that's unlikely to be the actual target. So I'm just seeing in the chat here, I know we have a question time at the end, but since it's related to this slide, CT and CQ are often used interchangeably. CT is cycle threshold. CQ is the technically more correct term. It's cycle of quantification, but again, because these are not quantitative results, it's a bit misleading to use, but they are used interchangeably and are exactly this same value. So it's that point where you've got this threshold crossing. Okay, so The CQ or CT values are influenced by a number of factors. So this can be the actual PCR kit that's used. So if you ran several different test kits on the same sample, you may get a different CT value. It may depend on when the sample was collected. So if you tested the same person at different points in time, you may get a different CT value. It can sometimes be impacted by the machine that's actually used for testing, again, because that can affect how the threshold is set. So you want to make sure that you are using an instrument that's validated for the test that you are using, and that you understand if there is an impact on that cutoff CT value or not. May not be, but it's a possibility. And then the technique of the health professional in obtaining the sample / the sample type. So again, that's just relates back to using an appropriate sample method. So for example, when you test someone with a saliva collection at the same time as a nasopharyngeal swab collection, you may have a different CT value. The idea is that, you have a range of CT values that are still going to give you an accurate representation of what a positive sample should look like for a specific sample type. So it is important to factor that in terms of ranges and cutoffs and interpreting the results that come out. And one important note is that while generally, depending on that sample input and with all else being kind of equal, an earlier CQ value or CT value would indicate a higher viral load of the initial sample. They don't actually tell you how severe of an infection you have, and certainly not if you're more likely to develop severe disease. So there's a lot of other factors involved there from just a clinical perspective. So they can't on their own give you any of that kind of clinical information, but can suggest trends and can suggest at least positivity, which is the main goal for these tests, is a positive or negative output. So one other aspect of PCR testing, and in particular, quite a few of the COVID-19 tests have what's called multiplexing capacity. So what that means is that you are multiplexing or putting together multiple gene targets simultaneously in one tube. So instead of having to run multiple assays to look for each target separately, you can run them all together. And on a very basic level, what that means is that fluorescence output that we talked about in the last couple of slides here. Basically, you'll be looking at a different fluorophore, different fluorescent marker for each target. And the instruments, if they're set up to do multiplex detection, will actually have a different filter that reads that fluorescence in different, what's called channels, essentially. So they're set up for wavelengths of excitation and emission to separate out those fluorophores from each other, so that you can look at 3 or 4 or 6 fluorophores at a time, just depending on the instrumentation and the test design. So with running multiple assays, there can be variations within each run, which could impact results. So if you're using a multiplex test, you want to pick one that's been validated as a multiplex, or if you're going to do your own kind of combination of tests together, you want to make sure you have some validation protocols in place to make sure that combining different targets is not going to give any erroneous data when you're actually going to run samples. So what this multiplexing means, as I mentioned, is it just requires that you have a different pair of primers, so those kind of endpoints that target your region of interest, or your target of interest, and then a probe for each of those targets with that different fluorophore on it. So the advantages of this testing is that it can be quite cost effective. You've got fewer consumables, and it saves time, mostly due to less prep time, but also 0.3 there related to higher throughput. So instead of running 2 or 3 tests per sample, you can run 1. In theory, that can increase your test accuracy because you are not having to look at, well-to-well variation within assessing for a single sample. So you kind of can do it all together, and you get more information with less samples. So you have less work basically cut out for you there. And for variants, we wanted to touch on this just a little bit around how kind of genetic mutations might affect PCR testing in general, and in particular for COVID-19. Obviously, this is something that has been pretty thoroughly explored. So what can happen is that obviously viruses, as with everything else, does mutate over time. In particular, they can mutate quite quickly. So in comparison to some other PCR tests for more kind of stable organisms, this is one where, we're having real-time rapid changes that kind of are quantifiable and at the forefront of people's thinking. So a couple of things with this. From FDA, we know that tests that detect multiple genetic targets are less susceptible to the effects of genetic variation than tests that detect a single target. Basically, you're just hedging your bets because it's very unlikely that you're going to have a mutation in both targets at the exact same variant. So it doesn't eliminate it entirely, but at least you will have a situation where you're getting one target positive. So how that works is, if you kind of have a look at the diagram to the right here again, those primer regions are designed, as you can kind of see, to sort of be complementary to those single-stranded portions, and like I said, kind of flank the target region of interest. So what can happen is that if there are mutations that happen to be in those areas where those primers bind to, or in particular in the area where the probe binds to, it means that that binding either is not going to happen or it's going to be happening at a less efficient rate. So you might be getting much later CQ values or CT values, or it means you're going to be getting negative test results when actually that target is there. It's just a mutated version of the target. So to give you some context, the viral genome for COVID-19 is about 30,000 base pairs, so pairs of DNA bases. The primers and probes are in the range usually of about 20, 25 base pairs. So it is a very small portion of the genome that would have to be changed in order for this to, or this being a specific viral mutation, to actually affect the performance of a specific test. Now, that said, it's not an impossibility, and tests should incorporate a way to screen for impact to their test by looking at new sequences from new variants to see whether these mutations are present in these primer and probe binding sites. So what that means is that, as kind of the first point on this slide mentions, we want to consider that potential for future viral genetic mutations, both from the design perspective, in the sense that you can choose regions that are maybe less likely to undergo mutations because they're not under the same kind of selective mutation pressures, but also kind of in an ongoing perspective from selecting a test, it's possible to look at whether there is a plan for ongoing post-market analysis for tests to incorporate analysis of new sequences and new information. And that's particularly critical for an emerging situation like with COVID-19. So as of February of this year, FDA has only identified a few tests that are actually impacted by these variants so far, which is great. The variants of concern are mainly affecting spike protein. So in particular, if you've got a test that you're considering that has spike protein as one of the targets, it becomes even more so important to make sure that that's being looked at on a regular basis. So for shipping and storage handling, there are different options for this. Certainly, lyophilized or freeze-dried assays increase the robustness and reliability, and if they're packaged as individual tests, that also reduces the risk of contamination, kind of going from bulk reagents. So these assays are typically stable at room temperature. You're not looking at thawing or additional equipment for storage and cold chain requirements, so cost and space requirements can be reduced. And then there's some time savings, and I know we're going to get into this a little bit more in terms of our triple luck product specifically, but there's some time savings for not having to go through the process of thawing and calculating and mixing for different master mixes for what you might be looking at for some wet tests that are available. So that's just one factor to consider is, what are you equipped to handle from a storage capacity. So this this next set of slides, I'll say, just in case, is looking at ways to streamline lab processes and kind of what that could look like, and some information on our triple lock test specifically. So our Precision Biomonitoring SARS-CoV-2 RT-qPCR test is a multiplex test. It is qualitative, not quantitative. And it is, the form factor is a 96 well plate with lyophilized beads that detects RNA of SARS-CoV-2 causing COVID-19. The intended use for this test is to detect novel coronavirus from clinically collected NP swabs stored in viral transport media, and that should be used for people who meet COVID-19 clinical testing criteria, and it is for lab use only, intended to be used by qualified laboratory personnel. We do have Health Canada authorization as well as CE marking for this test. So you have some photographs of what the plates actually look like to the left here. So you've got that 96 well plate with a fully lyophilized bead in each of the wells. So each of those beads contains all of the reagents that you need to run your sample. You just add RNA, give it a mix, and seal the plate and go. So some of the differences with our test compared to potentially some other wet tests that require mixing or cold chain storage is that this is room temperature stable. Our clinical test results have shown 100% sensitivity and specificity in terms of concordance to reference testing. We have 3 targets for each tube, so we target 2 viral pieces of the SARS-CoV-2 genome, and then 1 internal control for RNase-P to look at what happens when you have a negative result. So this helps mitigate the potential for false results, false negatives, essentially, where you think the sample is negative, but actually there's been some failure in either the reagents or the PCR cycling or the sample collection. So this gives you some assurance that a human sample was collected, that the extraction actually did extract human RNA, and that there wasn't any detectable inhibition from the sample itself. So these, as I mentioned, are pre-mixed and ready to go, which can help reduce errors and contamination associated with having to mix reagents. This helps eliminate a few steps, which can decrease costs and is scalable in terms of capacity, and will work in quite a few thermocyclers that can take a standard profile PCR plate. So again, what we're focused on is simplifying existing laboratory testing by making this process a little bit more streamlined with no cold chain storage requirements required. So we've got shelf-stable, pre-mixed, pre-aliquoted, and that RNA-P process control as well. And in terms of the time factors to consider, compared to a typical wet laboratory test, and we'll actually be going into a little more detail on the steps themselves in the next slide. Some of the things that you would have to do for a typical kind of wet test is you want to actually have to measure, calculate, and mix all of your primers and probes together. And that includes your controls, your probes, your primers, your actual master mix components. So you have to make that up yourself. And then there's, of course, the actual test time itself. So that doesn't change much, but the triple lock test, but just by virtue of its design, kind of can help mitigate some of the time requirements for some of these other steps. And just to give you an idea of what might be necessary for a wet chemistry test option, so this means something that's not already pre-mixed, also has 2 viral targets and internal control. You're going to be going through steps to take out and thaw primers and probes for each of your genes, mix and calculate each individual primer to those concentrations, take out your master mix, get that thawed, and then you're combining all those amounts together, mixing it up, and then aliquoting to plates yourself. Then finally proceeding to adding your patient samples and going through the process to get ready to run. For our triple lock option, we eliminate the vast majority of those steps because you're just opening your pouch, putting your sample in. So you're basically skipping down to step 16 of the wet chemistry test option to run. And then that is also basically in terms of timing and throughput for those wet chemistry test options. If it's not a multiplex test, then your yield is going also to go down because each sample is going to be needed to be tested 3 or more times, depending on number of targets that you want to interrogate. So in terms of where this solution may fit, the next few slides just kind of go into this in a little bit of detail. So in terms of throughput labs, this works in a scalable fashion and adaptable to quite a few different thermocyclers. So whether you're high, medium, or small throughput, this is something that could be potentially useful. It, as we've mentioned, kind of can help improve some efficiencies and capacity in the lab just for time-saving purposes, and can also be adapted for other testing. So at some point, if you're interested in running other tests, you can definitely do that beyond COVID-19. So for places where tests are being run every day, but throughput is a concern, the reduction in time for prep for these tests in the triple lock format can make quite an impact. And that can then translate to improved patient outcomes with faster test results and make that measurable impact in a community. And finally, for temporary labs, but still capable of high throughput testing that may have been set up specifically to deal with COVID-19 testing, that might be interested in a stable dry testing format that reduces some of the refrigeration storage needs. This can be a great option for those cases. So if you're thinking of field testing setups, remote locations where high throughput testing is still needed, this can increase margins per test pricing and help expand lab capabilities at these types of sites. So really, in summary, triple lock can provide a cost-effective and streamlined, simple process to address the need for fast and accurate testing for COVID-19. And we accomplish that by improving throughput and time for testing. We provide simpler tests that also reduce some risks associated with using bulk reagents. The portfolio will eventually be expanded for new assays that are currently in development, so that potentially can expand test portfolios and revenue streams for testing laboratories. And this is a scalable and highly integrated system that we're trying to provide here to allow for in-lab processing and production to scale based on needs, and working on compatibility with quite a few existing thermocyclers. So thanks so much, everyone, for your time. I think Carolina is gonna mediate, all you say, for the question and answer.
Yeah. So if there are any questions, please type them into the question box, and we'll get them answered for you. At the moment, there is one. There are 2 questions here. Do you provide tests internationally? So I'm not sure, Amanda, do you want to address that, or?
Yeah, sure. So currently we have CE marking, so we are able to provide this in certain countries in Europe as a result of that within the EU. And we are assessing from a regulatory basis other markets that we'd want to expand to. So if you have a question on a specific market from the international perspective, please get in touch with I know there's going to be some contact information at the end of the slide, and we can give you some targeted information on where we are, if we are considering that specific area.
Perfect. All right, our next question is, my understanding is triple lock technology is invented by Precision Biomonitoring, and Biomeme is the manufacturer of the products with this technology. Now for technical troubleshooting, where do we contact, or who do we contact?
Yep. So there are two tests that Precision Biomonitoring offers, two types of testing that Precision Biomonitoring offers. Our triple lock version is offered in this 96 well plate format for high throughput laboratories. We also import a test from Biomeme for portable testing that is lower throughput but higher portability. So there are two different types of tests that we offer for real-time PCR for COVID testing. So, but to answer your question on where to contact, for both of those, you would contact directly Precision Biomonitoring. So we we're an authorized importer for the Biomeme test, and we handle all of the troubleshooting and customer service in Canada for that product. And then triple lock is a separate product that's in that high throughput format.
Perfect. Thanks, Amanda. All right, our next question is there a need for an extraction step with the patient sample before running the assay?
Yes, this is not a direct to PCR format. So you do need to have RNA still extracted. What we've tried to do is create some efficiencies on the PCR test setup portion, but it still does require an extraction step.
Okay. All right, and the next question is, what would you say is the minimum number of daily tests necessary to make the cost of the machine feasible?
That is a great question. It really depends on the throughput that you're expecting in your particular kind of case by case, and what you're actually looking to accomplish with your test setup. So I would say that answer can vary widely, but it is something that if you do get in contact with us at the end of this, that we can kind of discuss on a case by case to figure out kind of what those number efficiencies would be in terms of, cost per test and of course, factoring in the hardware costs as well. So unfortunately, I know that's not a great answer, but it varies so much. So there's not really one that I can give.
Yeah, perfect. And this comes from Peter. So Peter, if you want to reach out to us via telephone or our email address or vice versa, we can reach out to you if you're okay with that. Aside from that, if you can just send us a quick email at sales@precisionbiomonitoring.com, we'll help answer that question for you for sure. And we have one more here. Does the assay work on other sample mediums such as saliva, throat swab, self-administered nasal swabs?
So it has been, sorry, I'm making sure I say the correct regulatory words here. So it has been validated only with nasopharyngeal swabs. That is the approved use that is authorized by Health Canada. That said, in theory, it will work in any sample where there is RNA present from SARS-CoV-2. And those additional sample types can be considered on a case-by-case with specific laboratories within the regulatory requirements, both federally and provincially, for the type of test center that you are. So there are some additional regulations that can be considered in making that decision. So it kind of comes down to if there's an effective sample prep step, it should in theory work with this PCR test. And then the regulatory aspects of it wouldn't then need to be addressed separately on a case by case.
Okay, perfect. All right, we've got more questions rolling in. So this one is, are the target genes the same for the triple lock and the Biomeme assays?
They are not. They are different. So Biomeme targets spike gene And ORF1AB, our triple lock test targets E gene and UTR, so untranslated region.
Great. All right, one more here. What would be the most common factor that would affect PCR tests to test false negatives or false positives?
So it's a good question. I think one of the slides covers this a little bit, but there's a range of things that could be impacting false negatives and false positives. From a false negative perspective, it would usually be something around the sampling. So whether it's the time of the sample, the efficacy of the sample that was taken, or the prep step. So if that was, supposed to be a positive sample, those are usually the things that will go wrong. For false positives, the biggest thing is probably thinking of laboratory or otherwise sample contamination with some non-target organism, or with SARS-CoV-2 specifically, whether that's like your positive controls that you're using or someone that's in your facility. So those are probably the biggest ones, but there's quite a few factors to consider.
Okay, great. One more question here. How is the performance of the test when used strictly for screening asymptomatic people? We see mainly travelers who need proof of a negative PCR to board the plane.
Right. So we have not validated specifically in terms of clinical data in our Health Canada authorization or instructions for use specifically for asymptomatic screening. That said, as we sort of have discussed through this webinar, RT-qPCR, real-time reverse transcription PCR, is really the gold standard for doing this kind of testing. And so it should be quite effective, as any other test would be for asymptomatic people, but we have not validated that claim.
Great, thanks. We have one more here. Does PBI recommend a specific thermocycler for the triple lock assay?
So it has been validated on the CFX96, but it is compatible with others that would need to be validated internally, again, within your own kind of regulations, both as an institute group and federal and provincial requirements. But the test plates can fit in quite a few other instruments and should be effective where the fluorophores are compatible.
Great, thanks, Amanda. So we have one more question in the chat here. Just a reminder, if you have any further questions, please use the question chat box. That way we can record the answers. The question here is it okay in terms of quality test results to run pooling sample instead of one sample?
That is a great question. Again, we have not validated this with pooled samples. However, we are working with regulatory bodies, so both Health Canada and FDA, to assess what we may be able to do for sample pooling. At this time, the test is not authorized with pooled samples, but again, depending on the specific regulations around where and how this test is implemented, that may be something that can be independently validated at your particular site. And again, we're trying to work on some recent changes that FDA has made to those requirements to see if they might be applicable here as well. So maybe more to come on that, but currently it's not under the authorized use.
Okay, great. One more question here in the question box. Do you have an idea of what the false positive rate is when actually used in the field?
So based on our clinical testing that was submitted to Health Canada, which is certainly a smaller number of samples, but representative of what we hope that performance will be, we did not have any false positives on our either our cross-reactivity organisms or our negative clinical test samples. We are still in the early stages of distributing this product, so we do not yet have that kind of post-market feedback from users on the test. We haven't received any feedback of concerns of false positives at this time, but again, it's sort of in the early stages still. So I don't have numbers or percentages that I can share.
Great, thanks, Amanda. Okay, I don't think there are any further questions in the chat or neither in the question box. So I think this kind of concludes this webinar. Again, if there are any other questions that you might think of afterwards, please feel free to reach out to our email address, that's sales@precisionbiomonitoring.com. Or reach out to us directly via phone. We want to thank you for spending your lunch hour with us. I hope this was a webinar worth listening to. We're always very happy to see all the attendees come on board. So thanks again, and we'll be definitely hosting more webinars, so take a look out for next month on a different topic, and hope to see you very soon. And Amanda, thank you as well for a great webinar, as always. All right, everybody, I'll be ending the broadcast with this. Have yourself a wonderful day. Bye-bye.
Bye.